All right, we're going here. So yeah, this is our last class meeting of the semester. Of course, you guys still have some work going forward. Let's see, obviously completing data collection, finish coding your scripts for group project 2 if you haven't, and then data collection. All groups will be collecting data from either 30 or 40 participants. And then depending on whether you have a between subjects factor or not, and then of course you'll be writing up group project 2, the paper. I sent some detailed information over an email over the weekend. I'll also kind of make a formal Microsoft Word document with most of that same information that I'll post on Blackboard. But I sent that email and I've been saying it in class. So you should have a very good idea of what you'll be doing for group project 2 as far as what my expectations are, though I will formalize that and I will post it here in the next day or two on Blackboard, basically reiterating the same stuff that I've been saying in class and sent in that email. And also we will have a test, much like the first one. You guys did very well on the first one, which will be, open book, open note, open article test. I do ask that you guys not communicate with each other during the test. So you can use any resources that you currently already have, but I don't want you guys communicating with each other during the test. I will be posting a study guide just like I did for test 1. I will also post that here. Well, I'll do that after I post some of the stuff on. The priority right now is to get some of that information regarding group project 2 into a formal document for you to read. And then after that, I will post the study guide. The test will be during so-called finals week, but you don't have to take the test at any specific time. You can basically take it whenever you want during finals week. But once you open the test, you have to finish it in that setting. So make sure that once you open the test, you are ready to finish test 2. So that's it. So today we're going to finish talking about, we're going through rather quickly in these last couple weeks of the class, the last few lectures of the class, we're talking about some physiological measures. Let's see, I guess we'll be talking about basically 5 of them. Last time we already talked about measuring heart behavior with ECG. We also talked about measuring blood pressure. And we talked about a couple of different findings regarding heart behavior. You read a couple of heart articles and you also read a blood pressure article. Today we'll be talking about measuring muscle behavior. And then we'll also be talking about measuring a specific type of muscle behavior, basically muscles of the eyes, and then electrodermal activity, which your final quiz concerns electrodermal activity. I want to point out now, and then I'll go into more details about it, I did find an error in the quiz that I made. It's definitely not entirely my fault, but the question that I asked, given the information in the article, was vague. And the answer was a little bit ambiguous. And so there's question number 1, which I'm going to I copied and pasted, or I took a screenshot, so I'll show you exactly the question. I'm going to make it so everyone gets credit for that. So people have already started the quiz for the EEDA quiz, so I can't make changes to it now. I'm just telling you that that everyone will get credit for this. I'm going to tell you, I'll show you the mistake, and then everyone will get credit for question number 1, whether you provided the answer choice of A or the answer choice of B as your answer. And so I will get to that in a second. So let's start here talking about measuring muscle activity with electromyography, or EMG, basically electrode sensors, which are placed on the skin, and they can measure the electrical activity of those muscles that are underneath those parts of the skin. And it's most oftentimes used for measuring voluntary muscles. I mean, technically, the ECG setup could also measure the EMG setup, which, oftentimes you'd measure like voluntary muscles, meaning like the muscles of your arm, or, muscles of your leg. Those are, some classic examples of voluntary muscles. Also the eye muscles are a little bit of, we'll be talking about those with respect to EOG, and those kind of fall. Some eye movements are kind of automatic blinking, whereas some eye movements are kind of voluntary. So when we're talking, though, about EMG, usually we're measuring voluntary muscle behaviors, though there's a couple exceptions. So We'll talk about these two aspects. So EMG is used in a lot of stuff, and I'm not going to do even a close to comprehensive coverage of the different types of studies that EMG has used. But like all these physiological measures that we've been talking about this last few lectures of the course, one of the things which is helpful about these physiological measures is they kind of help to provide information regarding behavior and the mind. Kind of a lot of the studies in psychology will use dependent variables that are self-report. Sometimes studies will use reaction time, which is what we did a lot in the first half of this class. These physiological measures that we're talking about in this last half of the class can kind of augment or provide kind of new information about behaviors, about things which are studied in psychology with purely behavioral methods. These physiological methods that we were talking about here can provide kind of a new perspective on something which, especially in the past, has been studied with purely behavioral or self-report methods. And so that's what I'm going to be talking about here with EMG. We'll be talking about two different ways of using EMG. So one concerns a theoretical question regarding hemispheric differences in emotion expression. That was the topic covered in EMG 1. And another, which you don't have an article for, but or you don't have an article quiz for, but I'll present some of the basic data from this study, some of the embodied mechanisms of emotional mimicry or emotional contagion, meaning basically the idea is that when other people are in an emotional state, they kind of express it, outwardly. And one of the ways, in theory, that we understand or maybe even empathize with other people's emotional expressions is that, or emotional states, is that those parts of the body which they are using to express their emotions, when we see them doing that, part of the way that we understand what they're doing is that those same muscles in us become slightly activated. So for instance, when a person is smiling, part of the way we understand that they're smiling is that some of the muscles that we use for smiling will become slightly activated. And that, in theory, helps us understand that another person is smiling. Similarly, if a person's frowning, those muscles that the person's using for frowning, one of the ways we understand that is some of the same muscles that we use for frowning will become slightly activated. So this is what I mean by emotional mimicry. We kind of one of the ways we understand emotions in others is by kind of subtly activating those same muscles in us that we would use to express those emotions. This kind of falls into a bigger picture idea in psychology and neuroscience related to embodied cognition. So those are the two things we'll be talking about with respect to EMG. So here's some background information. You probably were able to kind of figure most of this stuff out from reading EMG 1. It did have some problems, especially with respect to the statistical analysis. But one of the things I really liked about EMG 1 is that it uses kind of a classic way of setting up a theoretical motivation for a study. And so I'll kind of just get to the highlights of their introduction, the highlights of this theoretical motivation here on this slide. So it's known from previous research pretty clearly that smiling, it's involved this muscle, the zygomatic major muscle or muscles. There's muscle, it's kind of the muscle group because there's a left and a right side. So you can kind of consider it a muscle group. It's not really just a single muscle. And the zygomatic muscles are known to lift the face, especially the mouth, upward, which, as I'm sure you guys mostly know, I'm sure you guys all know, that is what you do when you smile. So zygomatic muscles become stimulated when smiling. And there's a different set of muscles that are activated when you're frowning. And the ones that are studied the most are these corrugator muscles. I believe this is pronounced supercilii, corrugator supercilii muscles. I think one of the things I think is interesting, though, is that the muscles that are the focus of sadness when you're frowning are not actually any of the muscles that are in the mouth. They're the ones that are right here, which cause your forehead to wrinkle. So when you're experiencing general negative motion, sadness being like the primary negative emotion, the thing you oftentimes experience is kind of the crinkling or the furrowing of your brow right here in a down direction. So these are kind of two muscle groups, the zygomatic and the corrugator, which map on to two different emotional expressions, happiness and sadness primarily. And so that's kind of the basic background information. Here's where we're getting to like the theoretical motivation for the study. And this is a very nice, simple kind of way of articulating a theoretical motivation. There's lots of ways of doing it. We did our own kind of way for group project 1. You'll each have your own ways for group project 2, but I just like this because it's a simple kind of way of theoretically motivating a study, and it's a common one. And if you do go into research after this, you'll have to write introduction sections, and this is a technique that a lot of people use, and that technique is to find 2 theories that currently exist on a given subject, which provide kind of different predictions regarding some phenomenon. And that's what they've done here. There's these 2 different theories. They call them hypotheses, but oftentimes in the literatures, hypothesis and theory, those 2 words are used interchangeably. So the 2 theories of the 2 hypotheses, the first one is called the hemispheric dominance hypothesis. And it's basically these are hypotheses that are basically trying to understand what regions of the brain are important for the communication and or expression of emotions. And the hemisphere dominance hypothesis basically says the right hemisphere is more important for basically all aspects of emotion than the left hemisphere. The hemisphere dominance hypothesis says the right hemisphere is more important for communication and expression of emotions. The valence hypothesis, on the other hand, makes very clearly different predictions. Says very different things about the nature of emotion. The valence hypothesis basically says that the left hemisphere is more important for positive emotions. Sometimes these emotions, by the way, are categorized as approach emotions. I'm adding this stuff about approach avoidance because I actually see this talked about in a lot of social psychology textbooks and courses. So I imagine that some of you are somewhat familiar with this concept of approach and avoidance. Approach generally thought to be positive emotion, avoidance generally thought to be negative emotions. So the left hemisphere, according to the valence hypothesis, left hemisphere is more important for experiencing positive emotions, communicating, expressing positive emotions, while the right hemisphere is more important for communicating, expressing, perceiving negative emotions. So these theories make pretty clearly different predictions about the types of behavior that people should show with their muscles when they are expressing different types of emotions. And as you probably remember from EMG1, people were making in this study they were doing. Let's see if I have that information here. Participants were asked to produce happy or sad facial expressions. And the researchers were interested in which sides of the face become more activated. So the muscles, so I talked about EMG can be measuring arm activity, it can measure leg activity, and it can measure these facial muscles as well. These are voluntary muscles that you have control over. This EMG study was measuring these muscle groups, the zygomatic and the corrugator activity, and it was comparing the activity of these muscle groups, kind of the muscles on the left side of the face to the muscles on the right side of the face to see if if there was a asymmetry as far as which side of the face becomes more activated when you are expressing these different emotions. And there's an important assumption about this. This is a study which is making trying to explain a phenomenon regarding the brain. But if you notice, there's no imaging in this study. There's no fMRI. There's no EEG. And so they're making an assumption that's based somewhat decently on previous existing literature that those muscle groups on the left side of the face are controlled primarily by motor regions in the right hemisphere of the brain, and vice versa. The right side of the face, those muscles are presumably controlled primarily by the left hemisphere of the brain, kind of this contralateral organization of the brain relative to the body that we've talked about now many different times. That's the assumption in this paper. And so they're using this idea while people are producing these happy and sad facial expressions. So happy would be positive, the sad would be negative, to look to see if there's these asymmetries in muscle activity. And here is the results of this study. Now, as I said, the statistical analyses were not even close to ideal. I've read them several times, and they're just, they make several mistakes, the biggest of which is I think I mentioned this in your quiz. I was angry. I think I actually called the statistical analysis garbage, and it was an emotional comment, but I stand by it. The statistical analysis in the study basically is garbage. As I said, the thing I like about it is, one, it's an example of using the thing I like about the study is, one, it's an example of a study which uses EMG to measure a very common psychology / neuroscience concept. Once again, the other thing I like about it, though, is that the way it's motivated theoretically in terms of these two competing hypotheses. I like that. And so I was weighing the fact the statistical analysis is bad versus some of these other things that I like about the study. So I'm just pointing out the statistical analysis was bad. It was bad because there's three factors here. And I think I mentioned them in your quiz as well. There's the side of the face, so left versus right. That's one factor, the side of the face. The other is the type of muscle, corrugator versus zygomatic. And the other is the type of facial expression, happy versus sad. So there's actually three factors, but if you look closely, they just did three separate one-way ANOVAs, which is problematic in most students leaving statistics, but almost certainly leaving a research methods class, know that that's not the appropriate statistical analysis. It inflates type 1 error, increases the likelihood of a false positive. Not to mention that the way the results section was written was just a little bit confusing. Nevertheless, some aspects of these results are strong enough that even despite their subpar statistical analysis, there's some things that you can still see about the results. I'm trying to remember if I put anything in the notes. Yeah, I do have some stuff in the notes. There's clearly more activity when people are happy. There's clearly more activity in the zygomatic muscles compared to the corrugator muscles right here. So people are happy here, and probably when you're just looking at these data and you don't have the appropriate statistical analysis, the way I kind of look at it is like, okay, this person's happy. What's their left corrugator doing as far as the amount of EMG activity, the amount of muscle activity? What is the left zygomatic doing? You can see when they're happy, the left zygomatic clearly has more activity than the left corrugator. The right zygomatic clearly has more activity than the right corrugator. So the zygomatic, those are the happy muscles, are more activated when people are making these happy expressions. That is not surprising. Let's also look at what people are doing when they're sad. So we'll compare the left corrugator to left zygomatic. You can see the left corrugator is a little bit higher than the left zygomatic. The right corrugator is a little bit higher than the right zygomatic, but it actually wasn't clear from their results section because, as I said, the results section was not exactly ideal. It was not actually clear whether this difference was significant. It's pretty clear that with the zygomatic muscle group, that it's more activated when people are happy than the corrugator muscles. It's not as clear for the sad condition, where there's the trend is there, but it's not actually as clear if it's a statistically significant difference such that the corrugator muscles are more activated than the zygomatic muscles on the same side of the brain. But the major question of interest in this study, as far as those theoretical questions, the hemisphere dominance hypothesis and the valence hypothesis, is which muscle groups are more activated in general than which side of the face is more activated than the other for each of these emotions. And let's see here. In general, it is the left side of the face which is more activated no matter what what type of facial expression is being made. So we'll look at the sad. The left side muscle is more activated than the right side muscle for the sad. And that's actually true over here is for these right here. For the happy, the left side is a little bit more than the right side. Though obviously this is a small difference, but it kind of makes sense that there's a small difference here because we're talking about happy, and this is the corrugator muscles, which really aren't involved with happiness at all. If you look over here, the zygomatic muscle difference, pretty big difference between the left side of the face and the right side of the face. Basically what this is saying is when you're smiling, those muscles on the left side of your face that help you smile are doing more work. They're more activated than the same muscles on the right side of the face. And you do see kind of the same trend over here for the sad muscles once again. In general, the left side of the face, no matter what facial expression you are doing, whether it's you being happy, you the participant, or the participant being happy, or the participants being sad, the left side of the face was more activated. Remember that assumption. The left side of the face is controlled primarily by the right hemisphere of the brain. That's their assumption. And so let's see if so what this supports is the hemisphere dominance hypothesis, or at least that's what the researchers reported. They believe that these data support the hemisphere dominance hypothesis because whether you're making the happy emotion or whether you're making the sad a happy facial expression, the sad facial expression. The left muscle groups are more activated, which suggests the dominance of the right hemisphere. The hemisphere dominance theory makes the prediction that the right hemisphere is more important for emotional communication, emotional expression than the left hemisphere. And that is a reasonable interpretation of these data, and that's the one that the authors went with. Left side of the face is more activated for any type of facial expression than the right side of the face, which supports this idea of right hemisphere dominance for emotional expression of all types. That's EMG1. So here's another study which is kind of related because it's measuring, once again, these facial muscles, zygomatic and the corrugator muscles, with respect to different emotional expressions. But this is based on, so this is a study on emotional mimicry. Once again, this idea that one of the ways we understand the emotions of someone else is that we kind of recreate the emotional experience with our own muscles. So other person is frowning and they're sad. One of the ways in theory that we understand that we're sad is that we won't necessarily frown ourselves. So it's not like someone else is sad and then you start, someone else is frowning and you'll start frowning too. But there is kind of like a low level activation of those same muscles that we would use if we were frowning when we are observing someone else. And part of the idea, once again, is the activation of muscle groups is part of the way that potentially, theoretically, that we understand the emotional experiences of people out there in the world. And so, this has been found in plenty of studies, and I'll be showing it here on the next slide. In this study, they'll be talking about this one right here. Participants will be watching videos of people, and those people in those videos will be angry, sad, or happy. And I'll show you the results right here. There's the abstract if you want to read it. They have this mediation analysis, which I'm not going to be talking about too much. The reason they have this mediation analysis has to do with this claim they have. This is a claim about causality, that mimicking another person's facial expressions with your own muscle groups is the way you understand the person's emotion. They tried to use a correlational technique known as mediation to kind of provide support for that. I'm not very much convinced by their mediation analysis, so I'm not going to talk about it too much. Really, what I want you to understand is this general idea of emotional mimicry, and I want to show you the evidence that exists for that. So here are the data. The left, center, and right columns correspond to these different emotions. This is when you're viewing an angry face. This is when you're viewing a sad face. This is when you're viewing a happy face. And then here is the legend. I actually don't remember which muscle the depressor is. The ones that I want you to focus in here are zygomatic and corrugator. Once again, zygomatic tends to be associated with previous research on happiness, and corrugator, frowning, sadness, negative emotions. This one's positive emotions. And so I'm actually going to ignore the anger column altogether. And they really didn't find that much there in the anger column. At the beginning, I guess in anger emotion, when you're viewing someone, for the first 10 seconds of the trial, they showed a little bit more activation in your happy muscles, zygomatic. But then as you're looking at the angry face longer, the dominant one became, the dominant muscle was the corrugator. So there's kind of mixed results here, but it kind of looks like just noisy data. It doesn't really look like there's much going on here at all that you can be making conclusions about. These two, though, you can see that there's pretty clearly more separation amongst these different lines. When you're looking at a sad face, you see that the corrugator, which is the orange line here, the amplitude or the activity measured with EMG is generally higher than these other two muscle groups, particularly the zygomatic. But the reverse is true when they're looking at happy faces. There's a lot of activity in the zygomatic muscle group, and there's relatively less activity in the corrugator, in the corrugator muscle group right there. So this kind of corresponds to the previous study in the sense that, but it's different than the previous study. The first study is all the EMG1 study that you read was about the involvement of these muscle groups when a person is asked to produce these facial expressions. This study, though, is different in the sense that in this study, people are watching someone else make these facial expressions. But what you find out is that when you're watching someone else make these facial expressions, at least these participants were doing some of the same things that the participant that they were watching was doing as well. So the theory behind this is that potentially these types of activity, I've mentioned this before, might be causally related to our ability to kind of understand what someone else's emotion they're experiencing. And you guys maybe have heard of mirror neurons. That would be a region of the brain which might help support this phenomenon of mimicking someone else's behavior that you're seeing being performed. So yeah, that's it for that one. It's another EMG study. So now we're moving to eye. We have eyes and then EDA, and then we'll be done with physiological measures. So EOG is the acronym for this. Electrooculography. EOG measures electrical activity from these eye muscles right here. And these eye muscles help you do several things: move your eyes left to right, and then move your eyes up and down. These muscles presumably are involved, or at least activity from the eyes can related to blinks can also be measured with EOG. So lateral and vertical, horizontal and vertical movements can be measured with EOG, as well as eye blinks. And I'll talk about two different studies that kind of support these ideas. You already read one. EOG used EOG to measure eye blinks. This is the one where the readiness potential, the Bereitschaft potential study. EOG can also be used, as I mentioned, to study the movements of the eyeball itself. And so I'll talk about each of these. You read one of them already. You read EOG1. You do not need to know, by the way, the anatomy of these muscles or anything like that. So EOG1, this is the study that you already read. It measured the readiness potential. They call it the Bereitschaft potential, which is a synonym, which uses EEG, so electrodes placed on the scalp. And they also measured eye blink behavior, that is electrodes placed on the face, with EOG. And the data, this study was pretty darn straightforward, actually. I feel like when I wrote this quiz, I felt like this is one of the easiest quizzes I've written. It was really simple. You really just have to kind of read it and look at the data. And these questions, I feel like, pretty straightforward. So I guess my prediction is that the scores will be high on this quiz. The participants showed a readiness potential for voluntary eye blinks, but not for spontaneous eye blinks. So in case the difference isn't clear, a voluntary eye blink, and was the researchers like, okay, blink now, and then you blink. Or when they say sometime throughout the course of, in the next few seconds, I want you to blink voluntarily on purpose. Contrasting that with spontaneous eye blinks, which are the ones that we do hundreds or maybe even 1,000 times a day, which we're completely unconscious of, the blinks that we do to keep our eyes moist versus, using voluntary muscles to blink their eyes. And they were looking at the difference in the readiness potential between these two different types of eye behavior. When you're blinking your eye on purpose versus when you are spontaneously blinking, you're not conscious of the fact that you are blinking. So here are the data. Time point 0. These are all a bunch of different electrodes. F in the odd numbers, in case I mentioned this before, in case you forgot, the odd numbers generally mean left hemisphere. The even numbers generally mean right hemisphere. Z means the center, like right in the longitudinal section of the brain, right here. For this type of the readiness potential, usually they're most interested in like the central electrodes, particularly CZ. But they were interested in all three of these electrodes. There's a different figure, which I don't think I showed. Yeah, there's a different figure where they're really just focusing in on these three right here. At time point 0, you can see each one of these graphs right here has a time point 0. Time point 0 is the eye behavior of interest. Basically, at time point 0, that's when the person blinked either voluntarily or spontaneously. That's what time point 0 is. I believe one of the questions is the event-related potential that they're studying is that preceding this this behavior, or is it coming after this eye behavior? And you can see by looking at these negative numbers and the fact that the 0 is on the far right and then the ERPs of interest are to the left, that the ERP in the study, this is kind of the exception to the rule, most ERPs occur after a stimulus is presented or after something occurs. With the readiness potential, which we talked about before, the Bereitschaft potential, it's the stuff that occurs in preparation of a movement. It's the stuff that occurs before a movement. So voluntary, let's just focus in here on C3. Um, Voluntary movement is the dark line. The movement itself occurred right here, and you can see the readiness potential is that increasing negativity that occurs up to the point of the behavior itself, in which we don't see what happens after the movement. Presumably, this will go back to the baseline levels once that movement occurs. But for the spontaneous condition, there was no increased negativity. The spontaneous condition, the electrical activity in this electrode up to the point of the spontaneous eye blink remains right around baseline levels. Same thing over here in CZ, although it is notable that the amplitude of this actually looks a little bit smaller than it does in the left hemisphere. This was surprising to me because textbooks usually say that the readiness potential is strongest right in the central electrodes. That was not the case here. It looks like the readiness potential was actually stronger in the lateral electrodes on C3 and C4, left and right hemisphere. You can see, once again, this is kind of almost like a mirror image of what's going on in C3. Strong readiness potential for the voluntary eye blinks, but for the spontaneous eye blinks, nothing. There's no increased negativity up to the time point of the spontaneous eye blink. So that's EOG1. So here's an article that you didn't read for this class. I'm just going to try and explain it. It's another way of using EOG. Once again, measuring the muscle behaviors of the eyes. But this one's different, whereas the EOG study that you are interested in, they were using EOG basically to measure eye blinks. Here, they're with the studies on reading, they'll use EOG or another more recent technology, which is arguably more powerful than EOG, known as eye tracking, which is similar in some ways to EOG. It's just probably a little bit more advanced. Eye tracking is to measure lateral movements, and for reading, the most prominent lateral movements are those left to right movements that you make when you're scanning the words on the page. And eye tracking and EOG has for quite a while now been used to compare good readers, or typical readers. In this study, in this figure, they call them normal readers, to people that are, disabled readers. The name for usually is individuals with dyslexia, or another condition, alexia, which is similar to dyslexia. And they compare kind of some of the properties of the eye movements of people depending on whether they are good readers or not good readers. And some of those things are highlighted here, and I'll help you kind of understand some of the things which are shown in these figures. So it says here, disabled readers tend to make more backward saccades. So a saccade is basically just an eye movement where your eyeball moves from one point to the other, kind of in a straight, like some kind of, intentional movement from one place to the other. And disabled readers make more backward saccades. So when you're reading, usually you're making forward saccades. But presumably, if you miss a word, if you read it and you don't know what it was, you get to the end, you have to go backward and read it again. And you can see that here in the disabled readers, like moving forward is moving up in this graph. So this moving forward, like from left to right, is moving up in this graph, whereas moving down is moving back to the left. And you can see, for instance, right here, forward readers move to the left, but this is probably like the end of a page right here. So the good readers, they're reading and they're going right, left, right, and it keeps it going to the right, to the right, to the right, until they get to the end of the page, and then they have to go back to the left again, which is what that is. So that big step down for the normal readers, that's like when you're going to the next line of the page, or the next line of a book or whatever. But if you look at the disabled readers, they have one of those too. So presumably this is when the disabled readers get to the end of the page right here. But you can also see where these arrows are pointing. They're making these downward movements right here. And there's another one kind of right there. And then like another one right there before they get to the end of the page, which is presumably right here. So those are those backward saccades, or sometimes they're called regressive saccades, which seem to reflect having to read the word again. So that's not a surprising thing. If you're not good at reading, it makes sense that you'll have to engage eye behaviors, which allow you to try and reread the word. And that's what backward saccades are. There's a couple other, they make more forward saccades as well. So if you're just counting, each of these little steps is considered an eye movement. So usually we make kind of stepwise eye movement when we're reading. We do them really quickly, so we're not aware that we're doing all this. But if you count the amount of steps that are required to read a single word, people who are normal readers will usually use a small amount of saccades. They might like do one or two saccades and then they're through the word. whereas disabled readers tend to do more saccades. Part of the reason, though, is because they're going forward, then they're going back, and they're going forward, then they're going back, and they're going forward again. And so that causes them to just have more saccades in general. Disabled readers, individual dyslexia, more eye movements in general, whether forward or backward, probably reflecting, once again, the fact that they're having to read words all the time. One of the more interesting findings, though, is that the saccade amplitudes of individuals with dyslexia tend to be smaller. And so saccade amplitude, what does this mean? This basically means that, like, when you are reading a word and you have an eye move, as I said, you usually fixate and then you move your eye to the next part, and then you move your eyes again. When you do that, you tend to make big strides with your eyes. A typical reader, a normal reader, tends to make big strides. They'll go from one part to the next, and there's like their eyeball moves a good amount. That's what a big saccade amplitude represents. Disabled readers, on the other hand, they're making small eye movements kind of all the time. So instead of kind of a big swoop through the word, which is what most normal readers do, they're kind of looking, it appears, one interpretation is they're doing like a letter by letter type of analysis. You kind of get the whole gestalt. You kind of get the whole word, all the letters kind of simultaneously. Whereas individuals with reading disorders, they have to take kind of small little steps, small little eye movements through each word. And I have data from this study right here. So here's the number of saccades right here for controls compared to individuals with dyslexia. And the number of rightward saccades, you can see this is forward saccades. You would get presumably similar data if you're counting the amount of backward or regressive saccades as well, regressive or backward movements. And in general, people with dyslexia made more in this study forward eye movements, more forward saccades, consistent with what I told you on the last slide. That was particularly true for words that were long. So for long words, controls generally will make more eye movements when they're reading a long word compared to a short word. But the difference is not that big of a difference. So with controls, they'll have a little bit more rightward eye movements, a little bit like higher amount of rightward eye movements for long words compared to short words. There's the difference right there. The difference in height here, a little bit higher amount of more rightward saccades when the word is long compared to the word is short when the word is short. But if you look at the difference here for the individuals with dyslexia, they're making a whole bunch of forward saccades when they're reading these long words. They make a good amount when they're reading short words as well, but they are particularly have problems, are spending more, doing more rightward saccades for these long words, which kind of makes sense. Because once again, they're breaking down kind of the word into these letter by letter analysis. And if it's a long word, long words are going to have a whole bunch of letters, whereas controls tend to kind of get all of the letters kind of like the gestalt, kind of simultaneously process all of the letters. And so in general, controls have less forward saccades. Individuals with dyslexia have more forward saccades, especially the longer the word tends to be. So that's that first graph. Figure 2. This is saccade amplitude. That is like the size of each eye movement that you make. The controls generally are making bigger amplitude eye movements. And as I said, this is consistent, this idea that when controls, they see the word, they can kind of get one swoop through the word, and they can kind of get all the letters. Once again, the amplitudes are a little bit bigger for long words with controls. I guess for this graph, the thing that matters the most is that in general, the amplitudes of the saccades just tend to be bigger in controls. And this reflects the fact that they're able to kind of one long saccade or maybe two kind of medium-sized saccades can get all of the information out of each word that they need. With dyslexics, on the other hand, they are always having to use kind of this letter by letter approach. They make an eye movement for letter 1, then they go to letter 2, then letter 3, letter 4. And that results that the amplitude of each eye movement tends to be small with individuals with dyslexia. And it's interesting that it doesn't matter whether it's a short or long word with people with dyslexia. They kind of have the same strategy. Once again, this letter by letter approach, which results in the average size of each saccade, the average amplitude of each forward saccade being small with individuals with dyslexia. So this is a physiological measure of, and it's pretty strong. I've seen several studies which basically show the same thing. They're pretty clear eye movement correlates of reading impairments. And these are some of the ones that are studied the most, the number of saccades and the amplitude of each saccade that is made. All right, finally, the last physiological measure is electrodermal activity, or EDA. And a couple little just basic things right here that weren't really talked too much about in the article, but are important, a little bit important to understand. So, you know, this EDA, electrodermal activity, is usually measured from an electrode which is placed on the palm of your hand. Actually, usually it's placed like on the finger. And it's measuring electrical, one of two things, either electrical resistance or electrical conductance, which are kind of inverses. If you've taken electrical physics, then you probably are pretty clear with the difference between resistance and conductance. You don't really need to know the difference for this class between electrical resistance and electrical conductance, but it's good to know that that's what's basically being measured in electrical dermal activity, are these two concepts, and they are inversely related concepts, conductance and resistance, such that if resistance is high, that means conductance is low, and vice versa. If resistance is low, then that means conductance is high. So they're inversely related. And any study usually has one, they'll either say they're measuring conductance or they're measuring resistance. But you know, they're basically the same thing, just mathematically they're inverses of each other. Some couple things about, you know, why your palms get sweaty. One, it's thought that it can protect your skin from injury. It's probably evolved somewhat to help you grab objects. But we're mostly interested in talking about these EDA, in how do different types of stimuli that you might look at, or different types of contexts, you know, types of social situations that you're engaged in, how does that affect this kind of sweat behavior on the skin? And it's pretty clear that different types of emotional states correspond to different amounts of sweat being produced on the skin, and that can produce you know, measurable changes. And as I said, these two kind of approaches, which are inverse of each other, electrical resistance, which is measured in ohms, or the inverse of resistance, which is conductance. I believe that's pronounced Siemens, which is called mho. So if you notice, mho is just the opposite. So ohms is OHM, and then mho, which is the inverse, is MHO. So OHM, like backwards, that is mho, MHO. So resistance and conductance are basically inverses of each other. The study that we read, EDA, was measuring conductance, mho, right here. And then the question is how does this, in general, conductance, basically, if you have a lot of sweat on your skin, that supposedly makes it so electricity could propagate through from one electrode to the other more easily. So a high amount of sweat results in high skin conductance. Both of these are measured of EDA. Resistance and conductance are both considered types of electrodermal activity. The article that you read, though, focused in on conductance, which measured in mho. And here's a couple findings, and then I'll talk about the EDA-1 study. So there's actually been two studies that we've had in this class which have looked at, that have used electrodermal activity. We actually had in blood pressure 1, so that's an article that you read where the main theoretical physiological measure in that study was blood pressure. But I believe I've actually showed you the EDA for that study as well. If not, it's definitely a figure in this study. And they were looking at the SCR, and they were comparing men and women's responses to different pictures. And they found, you know, I talked about these findings, so I'm not going to talk about them too much. Again, basically, men and women had different responses physiologically to the different types of pictures. So men and women had different responses. They did not respond exactly the same for the 5 different types of pictures that were shown in that study. And I actually, the different types there, so there's one that was like sexually provocative, there was one that was neutral, there was one that was showing a threat. I believe there's a picture that showed blood, and one that had like a picture of sporting events. And they found, you know, different things. related. And I'm not going to go into the details of those again, but that's like a type of study where you show people pictures and then you look at, the presumably different pictures cause slightly different emotional states in these individuals. Some of these social emotional states might be moderated by your biological sex or your gender, such that men might experience more emotional responses to some type of an image than women. And there was some evidence of that in the study. Another study, this is talked about in the green textbook, which I didn't use, but it's kind of an interesting finding. And it kind of is good. It kind of some of the big picture things that we've talked about in different other studies throughout the course of this class, which is why I wanted to bring this up today. So this is a study on prosopagnosia, face blindness. The problem with the studies, they had very small sample size. Prosopagnosia is a pretty rare disorder of face blindness where you can't recognize people by looking at their face. And so usually if you find studies on prosopagnosia, their sample size is quite small. There's a lot of single case studies where the sample size is even one. And so they were measuring skin conductance responses in participants with prosopagnosia. And they were showing these people, the people with prosopagnosia, faces that they had either been exposed to before, that they had seen before, compared to faces that they had never seen before. And if you have prosopagnosia, it doesn't matter whether you've seen the face or not. You are not going to be able to recognize the face, and you are most likely going to report that you have not seen the face before. People with prosopagnosia have face blindness. And so even if you've seen the face before, you're not going to remember anything about it, and you're not going to remember seeing that face before. But one of the things that they found in this study is that people with prosopagnosia, even though they had said, I don't recognize this face, the people with prosopagnosia had actually seen the face before. But if you ask them, have you seen this face before? They'll say no. And I'll point out that is an explicit measure of recognition. It's a behavioral measure of recognition. You're saying, is this thing familiar to you? Have you seen this thing before? What is this thing called? Or what is this person's name? And the people with prosopagnosia could not answer that explicit task, that behavioral task. What's interesting though, and you do find these dissociations in the mind between the body and the mind, or the body and explicit behavioral measures pretty frequently. And there's some of the neatest studies that exist. Even though the people could not say that they had seen the faces before and they didn't recognize these faces, their skin conductance responses did differentiate between those faces that they had seen before and those that they had not. So it was bigger skin conductance responses for the faces that they had been presented before, even though they didn't recognize them, compared to faces that they had never seen before. So the reason that this is cool, or it's a neat big picture type of idea, is because they're using a physiological measure to measure something which is implicit, something that the participant does not necessarily have conscious access to. The participant didn't know that they had seen these faces before. And that's clear by their self-report behavior. They said, no, we've never seen this before. I don't know that person's name. But if you're looking at what the body, the skin conductance response in this case, the body seemed to know something that the mind did not know. And this is actually not the first study like this where this has been this is like the third study, I believe, where this has been kind of like a theme, or at least is mentioned in the study, where the physiological measures can show implicit or unconscious aspects of something that explicit, like self-report behaviors, don't get at. So some other examples, which we talked about a lot in the first half of the class, reaction time. Reaction time there's many, many, many different studies like this. We talked about, for instance, the implicit association test, which is we talked about the one for implicit association test for racial bias. We also talked about the shoot, no shoot measure of racial bias. Those are our studies that use reaction time. And what's interesting about those is that people show racial bias when you measure it with reaction time, like in the implicit association test. However, in many, many, many studies, if you ask these people, do you harbor racist, biases against people of different races? Most people will explicitly say, no, I don't. Where if you look at the reaction times in these tasks, implicitly they appear that they do have these implicit racial biases. Another example comes from blood pressure 1. And this is just something they mentioned in passing in the introduction. they suggested that blood pressure might reflect gender differences in emotional reactivity, that if you just ask people like to make an explicit response about something about their response to something emotionally, the explicit or self-report measures might not show differences between men and women. But if you're actually looking at physiological measures, sometimes the body, the physiology there will be detectable differences between conditions that will not be detectable if you're using an explicit self-report measure. So I'm presenting this, today. It's kind of a bigger picture thing. One of the reasons this class is all about physiology, and one of the reasons studying physiological methods are so important is because behavioral methods, especially self-report, that is explicit measures, have some faults. And sometimes you can access information about people that is unconscious or that is implicit using physiological responses. And in reaction time, not exactly a physiological response, but it has some things in common, is another one of these. There's several different examples. The physiology can make the distinction where sometimes the explicit self-report measures cannot. And then finally, getting to EDA1, this is it's a social psychology study, I would say, and they're using electrodermal activity. They're measuring, once again, skin conductance responses, MHO, to look at how people respond to different elements of eye contact when they're looking at a face versus looking at a control object, which in this study was a radio, which is shown, the word radios right there. So participants viewed the faces or radios, and the stimuli, either the face was either looking directly at the person or the gaze was averted. So that would be like my eyes, what they're doing right now. That's the direct condition, versus averted would be if the eyes were over there. I would say they were looking, how does this electrodermal activity, how does it change depending on whether the face, the gaze, is direct or averted. Another thing that was a factor that ended up, I guess it was kind of relevant, but the other factor is half of the trials were actually live faces. So presumably, they didn't explain this particularly well, but presumably for the live faces condition, they were actually looking at another person's face who was actually there in the room with them. And for the live radios condition, they were looking at a radio, which was in the room with them. And those two objects, faces or radios, are once again either directly facing the people or the gaze was averted. And in general, what they found is that the responses to the pictures didn't really do anything. There was EDA activity was not really modulated in any notable, significant way when you were just looking at pictures. However, when the participants were engaged with actual live objects, whether a face or a radio, there was some EDA activities, particularly the faces. Really, the radio didn't produce a whole lot of EDA responses in the people. The live face particularly did. And you can see for the live face, and I'll show you, I'm pretty sure I have the data here in a second, you can see the difference in the amount of EDA activity, that is the amount of skin conductance, depending on whether the face is looking directly at you, basically looking you in the eyes, or not looking you directly in the eyes. First, I do want to point out something. So I was looking through the quizzes, and I wrote kind of about this a lot in the notes. In fact, maybe I'll get those notes up here. I did make a mistake for this quiz. And basically for this question right here, I made a mistake. And I said that this was the correct answer when actually this is the correct answer. In my defense, this article was definitely, I would say, misleading about some things. And I don't want to go into it too much. I've explained what was misleading about the article down here. Maybe I should talk a tiny bit about the figure. So they talk about right here, the ordinates on the main graph. So this is a main graph, this is a main graph, this is a main graph, this is a main graph. The ordinates in the main graphs express the difference in EEG alpha power between electrodes F4, which is in the right hemisphere, and F3. And then this is an important part. Positive values indicate relative left-sided activation, and negative values indicate relative right-sided activation. So positive equals left activation. So that's a positive thing right there. So that means left activation. The confusing thing is that if you actually look at the inset right here, this is actually measuring amplitude. Activation is different than amplitude. And I made the mistake when I was making the quiz. And so I'm going to give everyone credit for this, no matter what you put, whether you put this or right now, people that put this get credit wrong. But I will manually go back and fix everyone. Everyone will get credit for this. It's basically, no matter what you put, you'll get credit for it. Though I don't want you to be extremely confused. So I want to try and straighten things out a tiny bit regarding this question. If you look, these are the actual alpha amplitudes, or the alpha power, rather. The black is the direct condition. And you can see that there's more alpha power in the right hemisphere in the direct condition than there is in the left. That's weird, though, because remember it says positive values indicate left-sided activation. And so if we're looking here, this is the direct, this is the dark bar, black, it's positive, which should mean left-sided activity. But if we look at these black bars, there's more activity in the right hemisphere. I'm curious to know if any of you guys saw this and were confused. Reasonably so if you were, because they did not do a good job of explaining this at all. The key notion here to understand what appears to be a contradiction, which I don't think this is actually a contradiction, the key notion, and this is something that we talked about way back in the day when we were talking about frequency analysis, is that when alpha amplitude is high in a given region of the brain, alpha amplitude is thought to be inhibitory. So when alpha amplitude is large, or large amounts of alpha amplitude, that means activation is low. And so that's kind of what this means. It's a little bit misleading because they're saying positive values indicate left-sided activation. What that means is that basically when there's more alpha amplitude in the right hemisphere, that does mean that the activity, that is the amount of activation, is bigger in the left hemisphere. It is false, though, and this is why this is not correct answer. It says for the live face stimuli, alpha power was greater in the left hemisphere for the direct gaze. That is completely false. And I was confused when I wrote the question. That's why everyone will get credit. The truth is alpha amplitude was bigger for the right hemisphere than the left for the direct gaze. But because alpha amplitude is inhibitory, that means that activation in the left hemisphere was more. So it's a little bit confusing. That's the explanation for this. As I said, everyone will get credit for this question no matter what. Ultimately, the thing that I cared about you guys knowing in this study anyway was the stuff about electrodermal activity. Once again, you can see that they're measuring MO, MHO, which is a measure of skin conductance. The inverse of that would be ohms, which is resistance. And it's the same stimuli right here, the direct versus averted gaze, for live faces versus picture. Or the gaze means it's the faces. And then the control object is the radio. Once again, for the pictures, they really didn't show much of anything. Not too much going on for the pictures. You don't really get a big EDA response when you're looking at pictures no matter what. When you're looking at the actual live objects, you particularly get a big skin conductance increase, a big response to the live faces. And that response is bigger when you're getting direct gaze compared to an averted gaze. So bigger skin conductance response is basically when you're direct eye contact compared to when the eye contact is averted. And you get that for the person, but you do not get a difference for the radio. So you get little boosts in skin conductance response when you're looking at a radio, but it doesn't matter whether the radio is making eye contact with you or not. But with humans, on the other hand, there is something special about eye contact, where that appears to engage your emotional system in some way more than when the person is averting eye contact. So something about eye contact does seem to engage us emotionally more than when the eye contact is averted, at least based on the amount of skin conductance responses, the amount of sweat which is produced on people's skin. And that is it. That is all of the stuff that I have to talk about for this class. We still have plenty of work to do. I want to make kind of a general announcements because I don't think I had a lot of errors like this for the quizzes. And when the one or two that did occur, I believe I caught them. However, I will go ahead and throw this out there that in general, if there are, I mentioned in the recent email that if you if if you have quizzes that you want to take over again, I will, in the near future, I'll be making it so you can take the quizzes over and over and over again. At that point, the quiz scores will be finalized. So all quiz scores will be in, but you can use these quizzes to study for the test. And you can, once again, take the quiz, see what you're correct, what your score is. And then you can figure out what the right answers are for these questions. And if you're taking these quizzes and you believe it's possible that there are other errors like this, I will offer extra credit to students that will email me, make an appointment, and kind of make their case, especially if it's convincing. If it's particularly convincing, that, I might give more points than if it's not entirely convincing. I don't think I made a lot of errors. I spent a lot of time editing these things to make sure that there weren't a whole lot of errors, but I feel bad that there were one or two errors. And so I want to give you guys the opportunity. If you think that there's a problem with one of the quizzes, I will reward you by being proactive and contacting me, and you could potentially talk about those problems. And especially if you're correct, if it turns out that I did make an error, then I will definitely give you a solid amount of extra credit points. But otherwise, in general, I want to encourage students to talk about their questions with me. So even if I didn't make an error, if you just email me and talk about a specific quiz question that you're unsure about, I still might give you some extra credit for it as well. I want to encourage you guys to talk to me and ask questions. And so that's my offer. I don't think there's too much else to say. I will be contacting you through email, definitely several times for the remainder of the course. I'll be announcing when I'm posting various things on Blackboard. There'll be several posts that are coming here this coming week. So definitely continue to check your email. And that's it. I'll stop recording now.